The overall goal of this procedure is to demonstrate an RNA gel retardation assay designed for the discovery and characterization of RNA-binding proteins. Application of this assay is used to investigate the interaction between TcdA and transfer RNA.
The Agarose retardation method can provide answers to key questions in the field of RNA biology. For example, whether a certain protein interacts with RNA, whether the interaction is sequence-specific, and the strength of the interaction. The main advantages of this technique are that it uses very small amounts of protein and RNA samples and yields very informative results about the protein-RNA interactions quite quickly.
Though this method can provide insight into protein-tRNA complexes, it can also be applied to many other systems, such as ribosomal RNA, messenger RNA, and any other kind of RNA molecules.
In the morning, retrieve a vial of frozen E. coli BL21DE3 transformed with the expression vector PET23D EC-UU. Streak a low-salt luria broth or LB plate supplemented with 100 micrograms per milliliter ampicillin. Invert the plate and place it into a 37 degrees Celsius incubator until colonies appear in the late afternoon. Inoculate one well-grown single colony in 5 milliliters of LB plus 100 micrograms per milliliter of ampicillin and grow overnight. The next morning, pellet the cells. Following centrifugation, replace the medium with 5 milliliters of fresh LB plus 100 micrograms per milliliter.